Good morning, everyone. Welcome to the RCN webinar. We're just going to let everybody trickle in here for another minute and we'll do our introductions and get started. We just have a few announcements that we'll make before we start the webinar. I'm going to go ahead and start with some announcements because that'll take a minute while everybody trickles in to the webinar today. Happy March 1st. I feel like this means it's spring or almost spring for those of us in the Northern Hemisphere. Today, we are very happy to welcome two speakers. So we have Joe Brown from University of North Carolina at Chapel Hill and Jolinda de Korne from Swiss Federal Institute of Aquatic Science and Technology, or EAWAG, as many of us know it. So thank you so much for the speakers for presenting today. Before I turn it over to Joe, I have a couple of announcements. So we have a journal club, webinar nominations, and an email list, and Kevin will put some of those links into the chat for you guys to see. And then just a note that for our April webinar, we have a presentation plan from Jingjing Wu at Rice University on measles, and Marina Mozgovaj, and I'm sorry I said her name incorrectly. I will learn how to say it, about some wastewater surveillance in Argentina. So we're excited about that. So enough with talking from me. I'll let Joe share his screen and begin. Just a reminder, the Q&A is where you want to put questions for the speakers, and the questions will be moderated by me or Kevin. So with that, Joe, please go ahead. Thank you. You know, we all do this so much.
I've been on so many Zoom meetings today. I mean, there's no excuse. All right. Hopefully everybody can hear me now. Hello. Thank you to the organizers. Thanks, Ali. This is I'm very happy to be here. Thanks especially to the people on the West Coast, because it's what, 8 a.m. over there right now. So I assume there's a west to east gradient of interest and motivation among the attendees here. So I'm hoping for some good questions later. So what I want to do today is really talk about environmental pathogen surveillance in LMICs, the complexities around it. I'm going to start out with describing some of the work that we've been doing in Mozambique. And then I want to go through a bunch of use cases that I think could be interesting to consider for this community. And then we'll do some, Q&A. So I've got a timer going so I don't eat into my co-presenter's time. First of all, I want to say thanks to everybody who supported this work. So a lot of the work in Mozambique that we do is funded by the Gates Foundation and is in partnership with the National Institute of Health in Mozambique, which is part of the Ministry of Health. And a lot of the data that I'll show today comes from my former PhD students, Drew Capone and Gauthami Rao. Gauthami's doing her postdoc with me now, and Drew is now at Indiana University. So Mozambique's in Africa. Hopefully you know where it is. And we came to this work around monitoring waste, not really through SARS-CoV-2 monitoring, although we ended up doing some of that there, but more around trying to understand how we could use waste streams as a way of monitoring the circulation of pathogens that are important in populations there. So we came to this through kind of an interest in supplanting stool samples as a way of looking at pathogen prevalence among the population. So one of the things that we're doing, we've been working there for 10 years in Mozambique with the Ministry of Health, and we've been trying to understand the impact of sanitation interventions on the transmission of important enteric diseases. So part of what we do in our studies there is we collect a lot of stool samples from kids, and we look at the presence and quantify pathogens in stool samples across kids who are living in a context of better sanitation conditions versus those who aren't, to try to understand how that affects things. And we do things like this, like we look across a broad range of pathogens and look at prevalence. This happens to be our most recent data from about 1,200 children between interventions, those kids who have better sanitation versus controls who don't. Controls are in purple and interventions are in yellow. And we see pretty big differences around a few things. So this is like hot off the press, really interesting work from our point of view in terms of impact assessment of infrastructure. But we also know that this part is really hard. So collecting stool samples and that whole chain around what we do with stool samples to measure pathogens in them is really hard. And so a few years ago, we started becoming really interested in, instead of looking at stool samples, to look at circulating pathogens to figure out what's inside latrines, and to ask ourselves whether we could instead measure pathogens in those matrices as a way of really understanding these things. So we use a lot of different methods to sample latrines, and it depends on the type. But you may have seen, we published this paper in 2020, which looked at comparing the detects of a broad range of pathogens between stool and latrine sludges. So for bacterial targets and for protozoan targets, what we observed was that the most prominent pathogens that were present in stool were also present in sludges from latrines that were matched with the stool samples. So the kids whose stool samples we're looking at were also using these latrines that we were sampling. And we observed that for bacteria and protozoa, at least, of the targets we looked at, that they were also present in the same rank order. So that was really interesting to us because it told us that if we want to measure those things specifically, then it would be fine and okay probably to think about using latrine sludges as an endpoint in our trials that we're using. So we didn't see the same, for various reasons, we didn't see the same rank relationship between the viruses and the helminths that we were interested in. But if you're interested in that there's a paper on that. Again, it came out in 2020. And that was really our first foray into looking at waste streams to look at pathogens. We've also done, in collaboration with colleagues at NC State and in Malawi, some methods development around how do you actually sample latrines to get a representative sample for pathogens. And that's also been published. We're still working on other studies that look at methods. So one of the things that we've been working on in this area has been around mapping pathogen flows in Maputo, because Maputo is the capital city of Mozambique. It's where our sanitation work is based. And obviously, we're really interested in the relationship between expanded infrastructure and function and how that can control pathogens. But zooming out from that specific question, we were interested in kind of how pathogens are flowing across the city, which is an interesting thing because in Maputo, like many other cities of low and middle income countries, the vast majority of urban residents are served by decentralized sanitation, so latrines of various kinds, not conventional sewerage. So in Mozambique, in Maputo, city of 2 million people, about 90% of the population are on on-site systems. So, and only about 10% are on the sewerage network. Other cities like Dhaka in Bangladesh, you can see here is 80% on-site. Some cities like Port-au-Prince, Haiti, for example, which I don't think is on this list, is 100% functionally on-site sanitation. Yeah, and so in the United States, it's about 80% of the population are on sewers and only about 20% are on septic systems. But it really, in low and middle income countries, that's the norm for urban residents. So in Maputo, there are a couple of conventional sewerage networks. They're in the main sort of older parts of the city that has more mature infrastructure. But then a lot of the densely populated areas, many of which are formal or semi-formal, are not served by those systems. So the formal system serves areas that look like this, sort of built up, proper streets and a grid plan and so on. But then a lot of where people live looks more like this. This is kind of the area where we're working, where these communities have grown up organically, and they're not served by conventional sewage. I don't know if you've seen things like this. This is called a shit flow diagram, or in more polite company, you might call it an excreta flow diagram. It's a map basically of where the sanitary waste goes and whether it's safely handled or not. And this is a kind of way of understanding where all the waste goes in a city for folks who are interested in urban sanitation like we are. And it goes across from, containment, how is this waste actually generated, and then how do those places get emptied and transported, and whether it goes into reuse and disposal. So we classify things into unsafely managed or safely managed in green. And this is the shit flow diagram for Mozambique. So what we were interested in mapping pathogen flows across the city was really in adding a layer of quantitative pathogen flows to this, to try and really understand not just where the fecal waste is going, but where the pathogens are going and to what extent are they safely contained. So overall, this assessment is around complementing our private domain pathogen sampling, like at households. And we wanted to look across rainy and dry seasons to really visualize what's happening with pathogen flows, so that could go into infrastructure planning and be a baseline for further studies of sanitation infrastructure expansion. For the data I'm about to show, we used the bag-mediated filtration system, BMFS, as well as grab samples. And we used our TACMAN array card that we've developed. We have developed and implemented one for stool samples, but we also did one that's an environmental card. And we can talk about later, if you're interested in TAC versus dPCR versus other methods. We're doing some work on that too. So in the data I'm about to show, we sampled across the city. I showed you that map before of where the sewage networks are. This is a map that just shows different sample types. So looking at soils, surface water, open drains, wastewater, and floodwater. We'll talk about this in a minute, but obviously when you're in a context that's unserved by sewerage and the waste is going across many different types of waste streams, then if you want to understand where the pathogens are going, you got to take a lot of different kinds of samples, and those things get processed in different ways. It's a whole layer of complexity that we don't deal with when we just talk about sort of conventional sewage samples. Some visuals from the field. This is a BMFS and a grab sample. You can see this is typical of drainage in the area. This is a drain that will serve important sort of hydrological functions in terms of managing floods and so on, and runoff. But also you can see this pipe coming in right here. This is a place where lots of wastewater goes to. So this is basically an open sewer that also functions as a drain. And I'd love to talk about open sewers later if you guys are interested. Just a lot to say on that. So I'll show you a few graphs just of what the data looks like. You'll see that, on the left axis, we've got log 10 gene copies per liter, and the sample type is across the top. So fecal sludges. There is a wastewater plant, so we sampled at the influent and effluent. And we also looked at open drains, floodwaters, surface waters, and soils. Soils are not here. And then we also overlaid whether this, again, because we want to parameterize the shit flow diagram for Maputo, whether it's safely managed all the way down to not at all safely managed. So, so some of this is safely managed, is on the safely managed side of things, and some of it is clearly not. These are the limits of detection. So you should look at the data above the dashed lines here for this particular assay that we're using. And this is crypto, Entamoeba, and Giardia, 3 of our protozoan targets. And so what you can see, let's go. here. I'll show you for a few different pathogens, what we can observe, some of the things that we can observe here. So for example, one of the things this tells us, just looking at interaggregative E. coli, is that some of the open drains have a higher density than wastewater. And generally speaking, it's sort of in this direction from left to right in terms of, the density of this particular pathogen target. For Giardia, one of the things that you can look at is between influent and effluent here, and noticing that what we observe is a reduction associated with the wastewater treatment process there. It's a very, very basic wastewater treatment process that's subject to all kinds of issues, but it's also one that appears to be somewhat effective in reducing this particular pathogen target. We also were culturing, and I'll get into the reason why we would do this, culturing FIB as well across these different types of samples. That's primarily because, when we think about wastewater monitoring for disease surveillance in a high-income country, one of the things that can be assumed is that wastewater is mostly being properly handled and treated and disposed of. But clearly in this case, a lot of the waste that we're interested in the places we're looking for pathogens is not safely managed, and it does potentially pose an exposure risk to people who live in and around the areas that we're working. So it's important to also look at the potential for viability of pathogens in these types of matrices, and that's something that we can do indirectly, at least through culturing FIB. So we do that as well. Here's int1, very high levels. Also, you see that sort of gradient across, from left to right. And then if you overlay all of this, the different types of samples and all the different targets, one of the things, it's just a way of kind of visualizing what all is there. And we did include some interesting targets here that might be a little bit, that might be a little different. Like we included HIV. These were no detects on HIV, but we included Zika virus over here. We had some source tracking markers looking at human mitochondrial DNA, and then a few other things on the helminth side that are kind of interesting, and a bunch of bacteria. All right, everybody loves this. Sorry about this slide. This is like so many different pathogen targets all at once. But again, you see sort of a left to right across the different matrices that we sampled. This is all Drew Capone's work. So I want to just, again, give him a shout out. It's a lot of work to put all this together. But it's, again, kind of interesting to look across the different sample types and what we're able to put together quantitatively. So you could imagine that, not only can we start to then look at using these types of data in understanding what exposure risks might be for people who are interacting with these uncontrolled waste streams, but also it can tell you, well, there are a lot of questions that we can ask about things using data like this, and I want to save a bunch of time to go through the use cases. By the way, we're also, one of the big things that we have been doing there is to compare BMFS and grab sampling with passive sampling methods. We are really interested in, more swabs and passive sampling methods. So we looked at the torpedoes as well as more swabs to look at whether we get similar data at the end of the day after using these different methods that are obviously have a lot of different characteristics. These are just some of our passive samplers in the environment. And this is from a forthcoming, this paper will be submitted soon, but we looked at across, all the different things that we're looking at: helminths, source tracking markers, protozoa, bacteria, ARGs, and viruses. Looked at this collapses a lot of data together, but looks at the sort of prevalence of our hits on our assays between these different sampling strategies. And we're going to be able to compare these quantitatively across all the different things we're measuring. For the most part, the BMFS, by the way, does a lot better with viruses, which is, I think that we expected that. On other things, it doesn't make as much of a difference. But this is all interesting to us because I think we're very interested in scaling this kind of work across low and middle income country environments. And to do that, we need to have, very scalable methods, right? And we'll talk about that a little bit. I'm going to skip this. We're doing a lot of experimental work as well to look at the relationship between more swab loading for different targets and the concentration of the thing we're trying to measure in the waste stream. So we use isotherms for that. All right, so I want to talk about some of the differences, in case some of you are not working in these types of environments, between high income country context and sort of low and middle income country context.
Joe, sorry to interrupt you. I want to give you like a 2-minute warning.
Okay, got it. Yeah, I've got a timer going right here, so all good. So some of the differences are that, most people are not served by wastewater. As I said, there's a lot more pathogens and a lot, potentially greater diversity of pathogens, completely different context when it comes to AMR. A lot more contact with animals, presence of pathogens, even in urban environments. So zoonoses and emerging pathogens are a big deal. As I mentioned, a lot of the waste stream measurements that we're doing, they do have exposure relevance. So viability can be important. And the global health stakes are pretty high because the burden of disease is higher. Lab capacities and focuses are different. In fact, I mean, a lot of low and middle income countries have really good culture-based and microscopic methods, more than what we've maintained. But also a lot of the lab capacities on the clinical side, very little on the environmental side. Sequencing capacity can be very limited. There's effectively no dPCR that I've encountered in most low and middle income countries. There are a lot of limits on supplies and reagents. So the whole context around what you can measure and how is different. Yeah, budgets can be really limited. The use cases can be a lot broader. And I want to, and maybe in the Q&A, we can talk through some of the use cases. The health and the data systems are really different. And also this thing about representativeness is a very different question when you have most people unserved by sewage. Just something to consider on South-North partnerships. So about half my work is in low and middle income countries. So I speak with some authority on this particular thing. The ethics can be very fraught on establishing relationships between folks who are from highly funded environments like us, like many of us, and our colleagues elsewhere. It takes a long time to develop trust and figure out what you can bring to a partnership with others. The funding landscape, super different. This work can be high risk and high reward. So you can do some really cool stuff, but it's also, there are a lot of ways that things can go sideways. I think of capacity building in two ways. So, a lot of our work with, the Ministry of Health in Mozambique has been around, helping them measure things in new ways and apply this data, but also they help our students and our folks understand, a totally different environment. So it's really kind of two-way capacity building, but you have to be intentional about it. There are a lot of different scientific contexts, and it helps to be flexible, creative, and humble. And it can be difficult and expensive. I think sometimes the perception is we're going off to do this weird stuff in other countries, and it's sort of like a vacation, but it's actually, it's very, very difficult to do this kind of work, in the places that ultimately could have the most to gain from the kind of generation of data that we're thinking about here.
Thank
So
you,
Joe. I guess I want to stop there. Yeah, and we can take questions.
Thank you very much. I really appreciate especially this last slide. So I didn't want to cut you off on it because these are really important points. We have a whole bunch of questions in the chat, and I'm going to be selfish and take a first question. And that's sort of, I'm curious how you're generating so much data. I'm wondering, like, what are your what are your views on like data access? Are you making these data open access? Are you sharing them through data repositories? How are you sharing the data, both with other academics, potentially like commercial companies or public health, and with countries like on site? And how are they involved in those decisions about data sharing?
Yeah, I mean, I think those are great questions. I mean, those are part of kind of the long-term goal of this particular work is around data transparency and having us do things that directly feed into our partner's mission, in our case, the Mozambican Ministry of Health, and how they would like to use and see this data. So on the publication side, I mean, yeah, we're doing all open access, everything. All of our data for studies like this get posted at OSF. So the idea is to have this freely available to whoever is interested. But again, to kind of feed into and work with our partners on how they can interpret the data that, in useful ways.
Thank you. Okay, I'm gonna get through some of these questions. So I'm just gonna randomly pick the order here. So did you include mpox in any of your work?
We're including it now. So all of this stuff was done, all of the actual samples here were taken about 18 months ago that you saw the data on. And we didn't include it in that card design, but that's something that we're doing now.
Great. Let's see. I have a question about the microbial source tracking markers, and there's a specific question about like what poultry associated marker you use, but and if you have that published. And then I'll just add to that, what sort of data insights did using those markers allow you to make, and like what was surprising perhaps about what you saw with those markers?
We haven't really got into the marker analysis too much. I mean, I think we used for we had a whole project, and actually this was published in the last year, I think, in ES&T around our use of microbial source tracking markers, specifically in Maputo, Mozambique. And most of them perform really poorly for various reasons. Like most of the ones that we think of, like HF183 and some of the other like avian markers, they tend to not be as great in this environment. So one of the things we were kind of interested in is including some mitochondrial DNA markers and some additional markers. I mean, I think on the chicken marker, we used one that's been published previously. We didn't develop a new marker for that one. People eat a lot of chicken here, so it's probably not telling us that much, around specifically like chicken waste, but we really haven't dug into this analysis.
Because of diet, like in
Yeah.
people?
Yeah. People eat a lot of chicken, so you're going to see chicken mitochondrial DNA show up in their waste, right?
Yeah. Okay, great. So there's a question about whether local public health agencies are poised to make use of the data.
Yeah, I mean, that's our main partnership here is with the Ministry of Health. So they're actually interested mostly in the cholera data that I haven't showed, but there was a cholera outbreak during the time we were doing some of this sampling, and we took separate samples for cholera analysis and used both TAC, but also digital PCR assays to look at cholera over time as that outbreak emerged. So they have questions around that. They're interested, I think, in the rotavirus data. I didn't pay attention to that there, but rotavirus we have vaccines for now, and the vaccines for rotavirus in Mozambique were rolled out in 2015. So it's actually pretty surprising to folks that we see so much rotavirus still out there. I think that speaks to the some of what we already know about the low seroconversion to the rotavirus vaccine in LMIC populations. But it's still, I think, a striking finding in a way. It sort of was, I think, interesting because it sort of held up a big flashing sign that there's still plenty of this circulating.
We're out of time, so I'm gonna thank you very much and ask you to stop sharing your screen. But I also want to thank you very much for your commitment to this type of work, because I know it's incredibly difficult and time consuming, and you have sacrificed a lot personally to travel so much away from loved ones for this work. So thank you so much for all of your hard work on this type of work, Joe.
Sure, thanks.
Okay, so Jolinda, we're ready for you to present now. Oh, and feel free, Joe, to answer some of the questions if you'd like in the Q&A.
Yep.
Great. So I guess you will see my screen and that you can hear me. Great. So hello, everyone. And thank you a lot for being here. I didn't realize that we would have so many people here. It's almost 100, so that is incredible. My name is Jolinda. I am a postdoc at the group Pathogens and Human Health. It's the group led by Tim Julian at EAWAG in Switzerland. And today I want to talk a bit more about method development that we have been doing over the last years. And this is mainly focused on digital PCR. And we recently built an assay targeting 7 targets simultaneously. So a 7-plex digital PCR assay to quantify respiratory viruses, but also have some process quality controls included in our surveillance program. So I split my presentation in 3 parts. I will first talk briefly about our surveillance program to give a bit of background. Then I will give some information about the digital PCR system that we have and how we multiplex and how we approach this topic of multiplexing. And then I will talk about the 7-plex assay that we recently developed and that we are about to implement in our program. So first of all, a bit of history. The Swiss Wastewater Surveillance Program started in 2020, of course, with the SARS-CoV-2 pandemic. And by that time, we started with surveying 11 treatment plants across, or they were mainly in the south of Switzerland, but also the 2 big ones in Zurich and Lausanne, close to Geneva. Over time, we made some changes in the treatment plans that we monitored because of the network building up, also because of budget. That changed quite a bit over time. So we went from 2 till up even until 100 in 2022. We had 6 treatment plans that we started monitoring in the beginning of 2021 that we have been monitoring all those years until now. So we have a lot of data from these 6 treatment plans. Those additional 94 were not all done by us at EAWAG. They were done by 9 different labs across the country. But after almost a year, we decreased the number of treatment plans to 44. And what we are currently doing is we monitor 14 treatment plans since July last year. And I think the plan is that we continue with those 14 treatment plans. They are quite nicely spread across the country. They cover the biggest cities. And for some of them, we also do chemical analysis. So for those 14, we monitor respiratory viruses on a regular basis, and we also do chemical analysis for 10 of them. So as you probably know, we have this surveillance program where we routinely monitor respiratory viruses. So we do SARS, and we have 2 targets for SARS, N1 and N2. And we also monitor influenza A, B, and RSV. We get water we analyze water 5 days a week for all 14 different treatment plans nowadays. I'm showing you here the data of these 6 treatment plans that we have been doing already for years. And we have all the data out there on the dashboards, and they can be accessed by this QR code. I don't want to go further into detail on this data, but the main topic of this presentation is this 7-plex assay that we've been developing. And for that, I first want to explain a bit how we multiplex using our system. We have the NICA 6-color system of Stilla Technologies in our lab. I'll get to that later. If you look at our dashboards on our website, you can see that we have been using different protocols. So there's also a bit of history there, which is probably familiar to you who have been doing a lot of, or who have been monitoring over time, that there's sometimes a change in protocol. And then you, of course, have to visualize that in a way on the website or on dashboards. So how we did it is we used this viral RNA mini kit until almost 2022. And from that point on, we changed our processing, our RNA extraction kit to our Promega Wizard and viral TNA kit. So we extract the total nucleic acids from the wastewater, and we've been using this extraction kit since almost 2022. And in our digital PCR assays that we've been using, there has been quite some changes. So we started with the 2-plex in the beginning of the pandemic, but over the last 1.5 years, 2 years, we've been multiplexing assays or multiplexing targets so that we can obtain a higher throughput with the same machines and the same number of samples. So until July, we had a duplex assay where we quantified SARS, first of all. That was the main goal. And then we also had an extraction control to control or to know more about our efficiency of extraction. When we started using the 4-plex assay that I just showed, we additionally quantified influenza A and B, RSV, and we added SARS-N2 as a target. And besides that, we run the duplex again to check for inhibition, so how much our sample is inhibited. And we do that by spiking in SARS-N1. And because SARS-N1 is already in our samples, we have to do this in another run. So we run each sample twice, once for quantification and once for inhibition testing. In July 2023, we were ready to implement a 6-plex assay where we quantified all the targets, so all the respiratory viruses in one assay, but we still had to run the inhibition assay separately. So we are still doing that now because we are spiking in with SARS-N1, which is in our sample. So we have to run the samples again to test for inhibition. And if samples are too inhibited, we have the rule in the lab that we then repeat them. What we want is that we only have to run one assay and that we can, in this assay, quantify, or with this assay, we can quantify all the viruses that we want to quantify, but also have our process quality controls. So we want MHV as an extraction efficiency control. And we added another target, which is MS2, that we are validating now to be used as PCR inhibition control. And this is a bacteriophage that is not in the wastewater, so we don't have to run the samples again. We can spike it in and measure them only once. The main goal of this 7-plex is that we increase the throughput because we merge the targets. We don't have to run all the samples twice. So as I mentioned, we have this Stilla technology system, the NICA system. It starts with these chips we use the sapphire chips of Stilla. They have different chips, but these are based on oil water emulsion, so the droplets are formed by that. We put our samples on the chips. The chips go into the geos, the thermocyclers, where the sample is partitioned and the thermocycling takes place. Then we scan the chips using a reader, and our reader that we currently use for the 6 and also the 7-plex assay is a 6-color system. So the reader has 6 different channels, fluorescent channels that it can measure, and then we analyze the data. So what is important if you multiplex an assay? There are actually maybe 3 most important things. So first of all, of course, you have to design the assay in silico, which includes target selection and primer and probe design. This is similar to single-plex assay. What is different for a multiplex assay is that the fluorophore plays an important role, because the clusters that you get in the end needs to be separable. So each target will give another signal in another color because of the fluorophore that you choose. And based on that, you can either separate the clusters, or it will be more difficult to separate them. So it's very important to pick the right fluorophores and the right combination of fluorophores if you build up a multiplex assay. Then how we usually go about multiplexing is that we first optimize the whole assay in singleplex, and then we start adding targets to the singleplex. So we build up the multiplex step by step, and we keep making sure that the concentrations that we measure stay the same. So they are always constant between the singleplex and the multiplex assay. So an example of how we would build up, or how it would look like if you build up a triplex dPCR assay, and probably some of you are already familiar with this. But for example, this, I don't know if you see my mouse, but these 3 background colors are the spectra of the fluorophores that can be measured by the reader. And then if you pick the fluorophores that correspond to the emission wavelength, you can have signals in blue, in green, and in red, depending on the target that you are detecting. And this is what below the droplet plots show how it would look like in singleplex. So you have a negative cluster, which is in the left corner down, and then the positive clusters are positive for one of the fluorophores. If you build it up, you will get single positive clusters, but you will also have droplets that contain both targets, or even all 3 targets. So you will get an exponential growth in the number of clusters that you detect. And you will have to, or at least with the software that Stilla offers, you have to manually say and define what each cluster is. So you have to pick the single positive clusters, the double positive clusters, and the triple positive clusters. This is, I'm not showing that here, but I'm showing that here in the next slide. So this is an example of a 4-plex assay that we used previously. So the one that detects RSV, influenza A, B, and SARS-N2. Let's see if I can spin it around again. The more clusters you get, the more dimensions you also get. So if you want to define what each cluster is, you need to plot it also in more dimensions. The more targets you have, the number of targets, or the number of clusters will then increase exponentially. So until the triplex, it's still kind of easy to define what cluster is what. If you go beyond 3 or 4 targets, it becomes a bit more difficult, and you're no longer able to visualize all the clusters in just one plot. Then I just want to show you how we built up the 6-plex digital PCR assay and which fluorophores we used and how this then looks like. So we picked a combination of 6 different fluorophores that covered very nicely the 6 different channels of our system. And they don't overlap too much. They overlap a little bit on the bottom, but it's important that the top of the graph does not overlap with the other one. Then we plot all these clusters that are then in this multi-dimensional space, because we now have 6 different colors. We try to plot them all in 2D plots to be able to define what cluster is what, and we call this process gating, which we have to do manually. And in these graphs, or these colors show the droplet clusters and how we gated them. So we, based on what we know about the clusters that we expect and where they should be more or less positioned, we defined which cluster is which, and we were able to gate them in this way. So this is what we currently run in our routine monitoring program. So then lastly, I want to talk about the 7-plex and how we go beyond 6 targets, even though we have a 6-channel system. So as I said, these are the fluorophores that we had for our 6-plex. What we added is this extra fluorophore here in that will show up in teal.
Sorry to disturb you, Jolinda. You have just want to remind you have 2 minutes left.
Okay, thanks.
Yep.
And this fluorophore targets MS2, as I said, and MS2 is we are aiming this to be our inhibition control. So just to show you how this looks like, it becomes quite complicated if you add a target, and if you have 7 different targets, meaning that you have 2 to the power 7 clusters, it becomes quite difficult to define your clusters. So we have been looking a lot at these 3D and 2D plots to see where the clusters are and which cluster is what, and we came up with those 2D plots that enabled us to gate, like to define what cluster is what. And yeah, we've been able to build this assay where each cluster is separable, so they are clearly separate from each other, meaning that we can gate them and that we can have a clear definition of them. And as I mentioned, we made sure that the concentrations for all our targets stayed the same after adding this extra target in our assay. So this is what I'm showing here. We compare our previous RESP6, which is our 6-plex assay, with the 7-plex assay, and we always want the concentrations to be linear, so to be the same. And yeah, this is basically what I show here. So for SARS, we had very similar concentrations, also for RSV. And it's during a peak season. One last thing that I want to show is what the comparison between SARS-N1 inhibition and MS2 inhibition look like. So as I said, we wanted to change to another target for inhibition testing. What we saw is that if we compare the values one-on-one, the values are kind of arbitrary, so there is not really a relationship. Didn't see any linearity or any other relationship. However, what we care about most is that in case a sample fails based on the inhibition that was measured with N1, we should also have it failing with MS2. And for that, we are at the moment gathering more data on inhibited samples to be sure that they are then inhibited for both targets, so that we are sure that we can change to MS2 for inhibition control. Just some take-home messages. We currently, in the Swiss Wastewater Surveillance Program, monitor 14 treatment plants, and we routinely test for SARS-CoV-2, influenza A and B, and RSV. If we talk about multiplexing digital PCR, there are many opportunities to increase the throughput and to have a more efficient process. And what is very important if you are working with a similar system is that the droplet clusters are always separable. With this color, sorry.
Yeah. No, just want to say time is up. So I think we should move to the Q&A session.
Okay.
Yeah,
I just want to
that.
thank the team. I just want to thank the team for all the work that they did. And this was definitely not only me. So thank you to the team, and thank you all for listening, and happy to answer questions.
Yeah, thank you, Jolinda. Before we move to the Q&A, I have a quick questions. So are there any advantages for the 7-plex dPCR assays that you want to highlight compared with other assays?
Sorry, any advantages for the
for the 7-plex dPCR assays that you want to highlight?
Yes, well, I think the most important point is that through multiplexing, you can achieve a higher throughput. And I think this is with digital PCR still an issue that, that is kind of compared to qPCR, we still have a lower throughput. So if we can multiplex the targets, I think this is the main goal.
Yeah, okay, thank you. So I have a few questions here. Did you adjust the primers or probe concentrations according to target copy number?
No, we don't do that. What we always I'm wondering, is that something that that you usually do if a concentration of a target is higher, you would lower the concentration of the primer and probes? Is that your question, if we did that?
Not sure about. All right,
Or is
so okay, okay, so we move to the next questions. If Elisa have further questions about that. So what is the throughput of your chip-based system? What is the throughput of what is the throughput of your chip-based system?
Of what system?
I think the question is the throughput, like if it's a chip-based system, can you run like 2 samples at once or 1 sample at a time?
Yes, the chips, we can fit 3 chips in 1 geode, and 3 chips means 12 samples, but that also includes a positive and negative control and duplicates. So we have 5 samples per geode, so per thermocycler, and also per scan we can run 5 samples, but we have multiple geodes in the lab, so we in total can run 35 samples.
Okay. Are there certain fluorophores that work better than others under different conditions?
Yes, I mean, we have some experience with fluorophores now that are kind of our favorites now because they just work well. Some we had a bit more problems also because of the combination of fluorophores sometimes give these issues with clusters not being separable. So I think it's mainly based on that. If we know that fluorophores can work together and they give good separation of clusters, then we stick with them, and we definitely have now favorite combinations of fluorophores.
Okay, so next question is, did you work on any software to analyze the output?
Yeah, this is a good question. We are definitely thinking about this to develop a cluster algorithm so that we don't have to do the gating or the cluster definition manually. And yeah, we are definitely trying to develop that to make it more robust and less manual. And yeah, but this is not yet in place.
And the next question is, how many replicates did you run for each sample, and how much variation did you see between your replicates?
Yeah, we run 2 replicates per sample, so we run everything in duplicate, and we see, yeah, we see a bit of variation. I think this is the short answer. Yeah, if you have any other questions about this, we can probably talk more, because I think this is quite a long story in itself.
Yeah. So the next question is assumption that singleplex is the right answer justified?
That's
That was
I don't.
my question, and just to be clear, I guess you justified, like you showed the multiplex is working okay because it agreed with the singleplex, and I was just wondering, is the singleplex result always like the right answer, the gold standard, or do you think it's possible that an assay could actually work better,
Yeah,
you know, with a different fluorescent molecule or in multiplex, for example?
that's a very good question. I think how we approach it is that we always validate an assay, and from that point onward, we trust our validated assay, and then we build it up. So we add another target, and we always compare to our validated assay. But of course, there are limitations on assay development, like in general, because yeah, how to know what the truth is always kind of a very difficult question, especially with digital PCR. How do you exactly know that what you're measuring is right? Yeah, but this is how we approach it. We always validate it, and then we compare it to what we validated.
All right, so I have one more question here. Did you test on different extraction inhibition controls before settling on MS2 and MHV?
MHV, that I am not completely sure about, but I think we, for MS2, yeah, we first tested N1, so SARS N1 as inhibition control. And yeah, I think inhibition is a bit arbitrary also because it's target dependent. So what we do is we set this threshold of 60%, which is again kind of arbitrary, where we say everything below 60% we should repeat it because we think it's too inhibited. And we are now trying to switch to MS2, and there we want to have this same threshold, and we want to have both targets corresponding, like giving us the same above or below the threshold.
I have one more quick question here. Can a multiplex assay be developed targeting RNA and DNA viruses or pathogens?
So both, oh sorry, I'm still sharing. Both viruses and other, for example, bacteria, or is that the question?
The question is, can a multiplex assay be developed targeting both RNA and DNA viruses or pathogens?
Ah, sorry, RNA and DNA. This, I don't think this is possible, because you would need a different thermocycling program, because you need this extra reverse transcriptase step for RNA. And I am not sure how this would work if you do that on a DNA target. But I'm not 100% sure about this.
Thank you
Thank
so
you.
much. Jolinda, for the great talk, and thank you everyone for attending the webinar. We'll see you in a month.